We have a professor of anatomy and neurosciences at the University of Melbourne and director of digestive science, which he holds degrees in physics and mathematics. He's a specialist in anatomy, physiology. He's received many awards. Also, the Australian government's Centennial Medal, the highest honor. And he is a specialist in gastro, and he has worked with his colleagues on the identification of therapeutical targets. He is also famous for having identified the intrinsic sensorial neurons, or sensory neurons. And we're very happy to welcome Professor John Furness. Thank you.
So I'd like to thank Olmix for inviting me to talk to you today. I'm going to present a lecture which is largely a review to try and put together something that is quite complicated. The gut has to react to its contents, and it reacts through many different ways, and how those reactions are integrated into output functions of the gut is one of the very, very most complex things we can probably try and talk about. So I'm going to just try to give you an overview of and illustrate that with some data so you can understand what we know and to some extent the extent of the problems. Let's go here. Okay, so this is our starting point. So oh gosh, sorry, wrong button. So as Michelle Neunlis mentioned, the intestine presents a very large surface, and that's a vulnerable surface. That is, it's a surface that is prone to injury and has at the same time to be highly permeable. So it's protective and receptive to nutrients. It's much larger than the total skin of the body, and what is inside the intestine is essentially external to the body. And it's a complex mixture of nutrients, of things that are absorbed with nutrients, of bacteria, of course, bacterial products, and also products of the body, the secretions, the digestive enzymes, and so on. The intestine has to have its nervous system. The intestine doesn't know what is going to happen. It has no knowledge of what you're going to eat, so the intestine has to measure and sense that and react to it sensibly. So it's not surprising that it has a sort of small brain in there. Now this is a little summary to try and tell you the sorts of information that has to be integrated and the types of signals. So here is the external world, full of nutrients and bacteria, chemicals that are the products of bacteria or come in with food, because when we eat food, we eat sometimes toxins, plant alkaloids, things of that type, and parasites, fungi. They're all in here, and they are detected by the cells in this very thin layer, the epithelium, particularly by endocrine cells. They signal to the immune system and signal to the external part of the nervous system, to the brain, to sympathetic ganglia. They signal by local and circulating hormones, and they signal to the intrinsic nervous system, and they also signal to release substances such as defensins, antibacterials. All of this has to be integrated, and that's our task, to understand this integration. Now, the major detector, the major sensor, is the enteroendocrine cell, of which there are 12 major types, but actually, if you divide it up properly, there are 20 or 30 types. The other part of this is large. This is the largest endocrine system in the body. And then there's the enteric nervous system containing very large numbers of neurons, and as Michelle pointed out, very many fewer connections to the central nervous system. A large number of immune systems, immune cells, and innate detoxifying defences as well, and around about 50 different types of receptors to detect what's going on and to signal to the immune system, to the nervous system, and to the endocrine system. So here's a summary of luminal receptors. I don't want you to think you try and remember all of these, that's impossible. But to just illustrate to you the classes and the numbers that are detecting information and information from which has to be integrated successfully. So there are a group of receptors here, which are the receptors for nutrients, essentially. So these include the taste receptors for sweet taste, for umami, for bitter taste, a large bitter taste receptor family, the sour receptor, and receptors, a range of receptors for proteins, protein fragments, and amino acids, and free fatty acid receptors, which are not only on enteroendocrine cells, but free fatty acid receptors are also, for example, on lymphocytes in the gut. Then there are receptors for phytochemicals, for small components of foods, so for spices and so on. And these are reactive, and I'll show you that later. There's some olfactory receptors, mechanical receptors, and receptors for very generalised properties like temperature, osmolarity, pH, and a range of pattern recognition receptors that are recognising molecular patterns, particularly in pathogens and bacteria. There are receptors for toxins that cause expulsion from the gastrointestinal tract, and an interesting group of receptors for advanced glycation end products, the components of food that is essentially overcooked. So this is the epithelium that we're so concerned about, and you can see it contains detecting cells, enteroendocrine cells, two of which are illustrated here, and very close, the nervous system, and in this niche here, also, of course, the gut immune system. So the major sensory effect is mediated through endocrine cells, but there is also sensory information detected by the enterocytes themselves, and these release hormones. So, for example, in this illustration, these cells have a range of receptors, even on the same cell type. So these are some of the major endocrine cell types in the intestine. I haven't even listed them all, but it just tells you what sort of cells they are and the sort of messages that they send. I'm going to talk a little about these cells in some. The ghrelin cells, they detect nutrients in the stomach, and they promote appetite and growth hormone release. So they are building the body. The gastrin cells, one stimulating acid secretion. I'm going to skip down some of these because I'm going to concentrate on this cell, the L cell, and this cell is, I'm going to use an example, and it will show you the degree of integration we have in the body. So this cell secretes GLP-1, which increases the sensitivity of the pancreas to glucose. So stimulation of this through this pathway is anti-diabetic because it increases insulin release and therefore assimilation of glucose. GLP-2 is also in these cells, and it stimulates mucosal growth and enhances absorption. I'll talk about that also as an example. And peptide YY from the same cell slows intestinal transit and reduces appetite. So these cells are complicated as a group, but even complicated as individuals, because they do, even an individual cell has a number of roles. So here's an example of how one of these cells works. So here we're talking about the cell in the stomach that releases ghrelin, a hormone. And this is a simple experiment, but an extremely interesting one. In this experiment, the animals have been given a bitter taste stimulant. And in some of the animals, the mechanism for releasing ghrelin has been broken molecularly. So when you give a bitter taste stimulant, the food intake goes up. That's because, of course, you're releasing this hormone, and if you remove the receptive mechanisms, then you don't get that increase. The message goes up, and if you remove the sensory mechanism, the message goes up more because there's no feedback. So this shows something very simple. It shows that a bitter stimulant, as you might take in an appetizer before a meal, actually does what it does physiologically, because what it does, it stimulates specific receptors in the stomach and releases the appetite-stimulating hormone ghrelin. So the fact that for thousands of years, even particularly in France, people take before their meal something that's a little bit bitter, it has a real physiological role. And this is what we have to try and understand. What is the real physiological role of different components of foods? This is another example. So in this example, we're looking at feeding with a sweet taste receptor stimulant. Now, the sweet taste receptors are on the L cells in the intestine, and when you use a sweet taste stimulant, and obviously, or carbohydrates are sweet taste stimulants, you get an increase in the transporter for uptake of glucose, you get an increase in the protein, and you get an increase in the function. And if you knock out the receptor that recognises the sweetness in food, then you don't get these effects. And these are quick effects. These happen within tens of minutes. So the gut is detecting the nutrient, reacting to the nutrient, and increasing absorption through a pathway that relies on the L-type endocrine cells. And other sweeteners also do this. So the gut then is reacting to its nutrients and changing its function. In the first example, changing appetite, and in the second example, changing the absorption of nutrients. And this, after a lot of work, is the pathway. So here is the receptor. This is a receptor that is tasting in the small intestine that you have monosaccharides, glucose and so on. It's causing the release of a hormone from the endocrine cell. The hormone is acting on a neuron. The neuron is releasing a transmitter, and that is inducing the gene and protein and function of the transporter that takes up the nutrient. Now, this is the enteric nervous system. I'm not going to talk about it, but just to remind you, this is another receptive part, and there are nerve endings in the mucosa that detect what's going on and react through these neurons to change motility, through these neurons to change secretion and absorption of nutrients. And Michelle told us all about that. This system has a system of very interesting detectors. The detectors are primarily linked to the endocrine cells, and that links to sensory neurons that go to the brain, sensory neurons that go within the enteric nervous system, two sorts, sensory neurons that go outside through sympathetic ganglia to the spinal cord, and other sensory neurons that go directly to the spinal cord. So I spoke to you about one example where nutrients affect nutrient handling, but there are other levels of carriage of information from the gut through the nervous system, which I can't talk about all of them, obviously. Now, so what I've tried to do here is to provide one case to give you a feeling for the degree of integration that is occurring. So this is the L cell. It has receptors for fats, for sugars, for protein breakdown products, and also for bile. And these cells are mainly in the distal small intestine, so they're exposed to the bile. They release GLP-2, PYY, GLP-1, and they act through the circulation, through the nervous system to the brain, to the nervous system locally, and they act on the blood vessels, they act on the Peyer's cells in the upper small intestine, act on the pancreas, and act on the barrier of the intestine, which Michelle talked about, and also on growth. So this is a complex, integrated system of control that we are really trying very, very hard to analyse properly. But the important thing for us in this audience is the nutrients acting on very specific sites to command the physiological changes. So the gut, I've talked about the gut dealing with the nutrients, but the gut has to defend itself, it has to protect itself. And that protection is multifaceted. It's protected through the mucus layer, through methods of toxin elimination, by vomiting, for example, by detoxifying mechanisms. So in cattle, in the rumen, the bacteria are detoxifying. In people who don't have a rumen, the detoxifications in the small intestine and liver enzymes. By destruction, by destroying things through things like defensins released from Peyer's cells, through the innate immune defences, and through adaptive immune defences. So all of these layers are there to make sure that the gut is adequately defended. So it's not a simple system, it's not a single system, it's a multiple layer of systems of defence. And when we come to the discussion of bacteria, which Hervé spoke to us about, it's a two-way thing. The bacteria act on the immune system, and the immune system acts on the bacteria. So we have a two-way talking there, so that the commensal bacteria can aid the system, and the system can defend against the pathogenic aspects of the bacterial load. So the mucosal barrier to defend the intestine consists of the mucus layer, in which there are embedded bacteria, and also the movement into that layer of particularly immunoglobulins from plasma cells, and also the barrier itself, the epithelial cells and the tight junction between them. So we've got several components: an active defence, largely passive defence through the mucus layer, and a defence through these living cells. So the active defence includes the release of enzymes from enterocytes, including the defensins, which attack bacteria, the activation of specific receptors that detect the patterns of activity in, or molecules in microorganisms, acting through the toll-like receptors and mighty 88. This is very interesting. If this transducer from the toll receptors is ablated, then you get colitis. So that shows you the importance of these effects, and you get a very, very severe colitis in mice if you take out this messenger here. And as well as the toll-like receptors, there are also nod receptors, and they also, particularly in the Panneth cells, cause the release of defensive molecules. So you have elements of passive and of active defence through the innate part of the immune system. Now, the barrier itself, so what we're talking about here, so these cells are integral to the problem, to the defence, and Michelle talked about gut becoming leaky. So here is an example. So here we have leakiness caused by heat stress. So heat stress is a very important problem in animals, in pigs and cattle and so on, in animal production. And we can see here that in heat stress, the permeability or the resistance of the gastrointestinal tract is decreased. So is in other words leaky, and now molecules can get across the gut, and if we use a fluorescent marker, we can see the movement of a fluorescent marker is very much increased in heat stress. So what can we do about heat stress? Well, we can do some things about it. I'm just going to talk very briefly about some experiments that we've been involved in. So here is a heat stress experiment, and we have here animals which are thermoneutral and animals which are under heat stress, and you can see this is a measure of permeability, leakiness. You can see on a normal diet, leakiness going up. This leakiness is clearly partly due to oxidative stress, and here we can see the reduction in this leakiness with a selenium-enriched food, and here selenium plus vitamin E. So leakiness can be protected against by food additives, but we need to do more work on this to find the most effective food additives for protecting this important barrier. So I talked about this very briefly before, but I just want to emphasise that when we're looking at the protection through innate immunity, we are reliant on recognising the bacteria and through pattern recognition receptors, or receptors recognising the damage to tissue and modifying the inflammatory response, and particularly modifying the production of cytokines that can regulate the inflammatory response and deal with the damage. Of course, we are often faced with the condition where there is an overproduction of the inflammatory mediators, and then we come into a pathological situation. The Panneth cells have pattern recognition receptors, as I said, and this shows the Panneth cells that contain the defensins. So they are recognising patterns through toll-like receptors and nod receptors, and they are causing the release of proteins which are defending and trying to maintain also the commensal bacteria, the bacteria which are helpful in the gastrointestinal tract. The adaptive immune response depends on the commensal bacteria activating tolerogenic systems through Treg cells, the production by those cells of tolerance to the commensals. So we maintain an equilibrium with our commensal bacteria, which we've done over evolutionary periods, but also inflammatory reactions where through TH cells we recognise the pathogenic bacteria, react to them, and have an inflammatory repair, but sometimes an uncontrolled inflammatory reaction of the gut. The antigen sensing I talked about there in very general terms, but that sensing is both through direct actions on receptors, but also by the recognition of antigen through M cells, so that they are sampling antigen in the intestine, communicating to follicular nodes in the gastrointestinal tract, connecting through there to mesenteric lymph nodes for the generation of appropriate gut homing T cells, but also some of the dendritic cells have projections through the mucosa, they react directly and they go back to peripheral nodes. So the integration is at the innate level at the mucosa and adaptively both in the mucosa and systemically. I won't talk about that, I think I'll go past that. So we then come to a situation where we see that we can have a healthy environment where there's an equilibrium between the commensal bacteria, the production of a defensive barrier, and a maintenance of that, and as Hervé talked about, a dysbiosis where there's a pathological reaction to bacteria that overwhelm the symbiotic bacteria. So these are pathobionts who will then damage the barrier, increase leakiness, increase the pathological inflammation. So our aim has to be to try and deal with the inflammation and maintain the barrier by adequate detection of the different classes of bacteria. This is an interesting thing because this is a illustration experimentally of what is going on. This is in this experimental work, the gut has been challenged and the inflammation has been measured by NF-kappa B. The challenge here is by butyrate and there's other challenges. And here, a bacterium which is protective, Delbrueckii bacterium is applied, and you can see the inflammation is reduced. So here we can see a commensal bacteria in experimental situation reducing inflammation in the gut. So we have a lot of knowledge and we have a lot of experimental data that this is true. So if this is a sort of conclusion in terms of our homeostasis. So the gut is always exposed to variable luminal contents and is always in a state of alertness, in a state of mild inflammation. The gut immune system and the bacteria have co-evolved over very, very long evolutionary timescales, and in healthy gut they're in equilibrium, and the bacterial, symbiotic bacteria keep a equilibrium successfully. And in dysbiosis is when this comes out of equilibrium and the gut immune system and the defensive system can no longer cope or can't manage the bacteria. So when we think of enhancing gut health for animal production, we have to think of using nutrient components that promote nutrient assimilation, give adequate animal growth because you promote assimilation, maximise production and carcass weight, use additives that are protective in the diet, so the diet components that protect by enhancing mucosal protective function, use of probiotics or manipulations of the bacteria also to defend and increase their gut health. And to put all this, we need a strong evidence base. That's what we have to try and get in the next little whiles. And coming back to my first slide, so what our challenge is to understand properly how all this integrates. So through the contents of the lumen, through their detection by around 50 different receptors, by those receptors activating immune, endocrine and neural systems, which also all talk to each other. So that's a challenge we have. We have a very strong knowledge base, but we need to take it the next step so that we can adequately apply this. So in English we say that when there's a lot of things together it's a can of worms, and is a way, but we have a can of worms, but hopefully we can work our way through that can of worms and get to a place where we can easily utilise our knowledge to improve nutrition and gut health. No, thank you very much.
Merci professeur Furness. Voici donc ces quelques images sous forme vidéo qui résument votre pensée, si tant est qu'elle puisse être résumée.
Being the primary organ of our digestive system, but also an ecosystem, an enteric nervous centre, and last but not least, the main organ of our immune system, the gut also provides the largest area of contact with the outside environment. These areas of contact, which in men or pigs can be as large as a tennis court, must ensure the proper supply of nutrients and at the same time manage the reaction to pathogens, toxins and parasites. The lights in the digestive tract are these sensors, which can communicate thanks to immunity-specific cellular receptors. They're everywhere, and they call lymphocytes and macrophages to their assistance.
Merci, vous pouvez applaudir.
Thank you very much indeed. A round of applause for this excellent educational video on our gut, this organ that controls us. Any questions for Professor Furness? No? Okay, let's take a break and let's be back in 20 minutes. You'll have coffee outside and we're back here in 20 minutes. Thank you.